So, the 100,000 Genomes project. In terms of policy history, this has come out of it's come out of a load of things that have been running over a number of years, particularly the House of Lords report for genomic medicine and the creation of the Human Genome Strategy report. That's kind of in parallel with efforts on the other side. You don't just need genomes to get them into the health service. You also need to be able to get access to the patient records that go alongside if you're going to do any sort of phenotype analysis. So all of the efforts to try and get the health records into a state where you can use them for research. And so that came into being effectively through the UK Life Science Strategy report, which funded CPRD, which is the primary care records, and set up the FAR Institute, which is going to build capacity around using records. A year later, the Human Genome Strategy Group report came out, and the update of the Life Science Strategy announced the 100,000 Genomes project. So in 2013, Genomics England was set up, and that's the organisation which is tasked with making this into reality. Genomics England is a lightweight organisation. It's less lightweight than it was. It's now about 40 people. It was about 10 last year. It's doing procurement. It's not going to build things. It's going to procure services to deliver this project. And I'll tell you what the state of that is now. So what it's got to deliver is quite a variety of different things. So obviously sequence 100,000 whole genomes from patients in the NHS. I think the key thing from the point of view of how people normally think of these studies, this is not a research project. This is a treatment project. These are all sick people, and they're going to be selected on the basis that if you sequence them, there's a good chance that by analysing their sequence, you can inform their healthcare. The research is a secondary thing that comes alongside, but there are other things to deliver as well, in the sense of obviously improve the health systematically, transform the NHS into using genomics on a wider scale, generate wealth for the country in being the first in the world to have this level of investment and to build up SMEs and expertise around using genomics on a large scale. And then it's a time-limited project. It only runs till 2017. To deliver this, you have to build infrastructure to be able to handle the data with respect to the health service. And so we're going to leave a legacy of infrastructure to be adopted by the health service. And hopefully, at the end of 2017, there'll be enough enough evidence that this is of value with the point of price for the sequencing then, that it will continue and it will reuse and extend the legacy. So it is big compared with anything that's been done previously. People will probably know about UK 10K, which is 10,000 genomes that was done at Sanger here. So you might think it's 10,000, it's 10 times bigger, but actually it's 100 times bigger because the amount of data being collected per genome is about 10 times more because of the depth that's being sequenced. And that's to get a genome, an individual genome, at the kind of quality where you have a good chance of being able to produce an individual interpretation. The actual specification is that 95% of bases must be callable for hets with high reliability. And if you look at the number of genomes available in the world right now at that sort of depth, it's very few. It's only hundreds to thousands in the Cancer Genomes Consortium, the International Cancer Genome Consortium, and there's a WGS 500 study in Oxford, which is kind of almost like a pilot for the 100,000 genomes in the sense it was, again, clinical samples. So it's very big. We've gone for whole genome sequence rather than exomes. I think there was a lot of debate about this about a year ago. I think it's now pretty much accepted that actually there's a lot of extra information to get outside the exome. We don't know how to use it entirely now, but if we don't collect it, we won't learn how to use it. And one of the things that you get from doing a whole genome, particularly in a PCR-free environment, which is what's being used here, is you get a very clean sequence. And so all this question about going to very high depths in exomes, you don't need to do, because that normally in exomes, that's just to compensate for the variable coverage you get when you do a pull down. So here you get a very clean genome sequence with high reliability. What can you get out of it? Well, the WGS 500 study kind of informs that. If you do rare diseases right now in the health service, maybe you interpret 15% of patients. If you do a whole exome, maybe you get it up to 25%. Doing whole genomes in the WGS 500, they have the impression that maybe 50% they have a good lead candidate of a variant that might explain the disease. And that's backed up by other publications. The Nijmegen group has published something very similar recently as well. So that's the sort of target that's being talked about. So Genomics England is going to target rare diseases and cancers, and they're going to be specified according to residual unmet clinical need. So you've got to have previous genetic testing if there is a genetic test for that. There's not going to be very much existing collections. It's really people who are in the health system now in need of treatment. It's going to be based around trios for rare diseases and based on informed consent. So there's freedom to operate in terms of doing research on this data as well as the interpretation and feedback. It's not just collecting a sample. There's a requirement to have the clinical data to go alongside that. And so as you'll see in terms of how that's collected in the health service, that puts certain conditions on data collection going forward. We're also going to collect not just a DNA sample for sequencing, but another sample which will be for additional research. So that's being collected in parallel. So the consent basically is to get a feedback for you as an individual, but also potentially use that for research as well. Cancer is very similar. There's a set of cancers that are being targeted. It's a normal and tumor sample. It's slower in cancer because of the question of how to deal with FFPE samples and whether you really can get the high quality sequence that you need from those samples. Feedbacks, this is the kind of difference which makes it a treatment project. There's going to be analysis run on each of these samples and fed back directly into the clinical genetics units. The NHS has been worried about the effects of that in terms of having a whole genome set of 4 million variants fed back. But the objective here is to analyze it from the point of view of the condition that the individual is in hospital for. So it's primary findings. It's only those findings that have been fed back. Secondary findings of a very small, limited number where we know there's clear high penetrance and actionability are fed back optionally if the patient wants that. The patient decides what degree of extra information there is. However, having said that, the information, the raw information, is also available to the NHS referring teams, and the patients can actually request their entire genome sequence if they want it. So you're protecting the patients from not having more information than they want, and the clinicians as well, but you're allowing them to take it away and analyze it third party if they want to. Now, so where have we got to? So we've done a pilot study. There's various pilots running. There's a pilot to evaluate contractors, which I'll mention in a minute. But there's been pilot rare disease running now for most of 2014, and there's getting close to 2,000 genomes, full genomes that have been collected in the pilot study. Now, if you think of the process to do this, you've got to go from sample DNA to clinical action. There's a load of steps. The way we're viewing this is a contracting view. So the sequencing is a step where you give a sample to somebody and they return standardized formats of data. Then the annotation to provide a clinical interpretation is another procured operation, and then that goes back to the NHS, as well as that data going into the database. We've evaluated 2 of those steps. So the sequencing was evaluated by giving samples to a number of different companies, and they came back and we decided that 2 of them met our threshold of quality, and then there was negotiation and the contract's been awarded to Illumina on the basis of that. And in fact, there's a building just around the corner here, which is being built by the Wellcome Trust, where the Illumina contract is going to be executed just through a rental agreement. But the point is that Illumina has to do this in the UK as part of this contract. So that's one of the basic principles behind all of this, that everything has to happen in England because it's NHS, it's Genomics England. In terms of annotation is obviously much harder, but we have gone through a process of filtering 30 groups who took part in a competition to evaluate a small number of samples and predict interpretation from them, and 10 of those are going to go forward to the next stage of running on a larger set of samples. So the state of the thing project is, oh no, I didn't want to do that, is sequencing contracts signed and running, and the building's being built and samples are being sequenced by Illumina. Annotation, we've got 10 companies selected to be involved in a second phase assessment. In terms of handling the data, the MRC has given an award to build a data center, which will mainly support research. And then for the sample collection, which is obviously one of the largest components, there's the genome medicine centers, which I'll come to in a second. The other side is research. So the research is the Genomics England Clinical Interpretation Partnership has been launched, and I'll mention that too. So genome medicine centers, you will probably have seen this. So this was announced at the end of December that 11 centers have been selected. These are the leads of those centers. So this is basically NHS England commissioning trusts. The trusts are bid to provide samples, designated numbers of samples from patients in their systems into Genomics England for sequencing. So that's basically up and running, will be operational by February. So this is the kind of picture that we've built up, that you've got the NHS over here, which will be the genome medicine centres. They can send patients, send samples to a repository, goes to Illumina for sequencing, and this comes into our data centre. Same with the information, phenotype information. Inside this data centre, these annotation providers will run apps inside a secure environment. They can't take the data anywhere else. It doesn't go and run on a website in California. It has to be part of the conditions, it has to run here. That'll feed back a clinical report into the health system for use, validation, etc. But that data there is also going to be available in an anonymized form for the Gzips partnership for research. And so we've now launched this call for bids from academics and industry to take part in that research inside this data center. So again, just like these annotators can't take data away, you can't take data away from here either. You have to run all your analysis inside that data center. So that's the structure. The Gzips, which have been launched, the bidding for this has to be in a couple of weeks. It's really to form groups that are going to analyze particular disease areas. So the structure up there is these are domains of disease. We want clinicians, academics to get together and form consortia to do analysis around disease areas. Also for some cross-cutting work around evaluating different methods, applying systematic pipelines, etc. So that's the deadlines for that. The expressions of interest have to be in by the end of January. So that's Genomics England. Pretty much everything to say about where it is right now. It's obviously now a very big project in the sense that it's coordinating these 11 genome medicine centers for sample collection, and it's potentially organizing, through these consortia, a consortia of consortia, going to be thousands of PIs essentially involved in interpretation within this environment. And hopefully it's going to lead to transformation of the health service in terms of adoption of genome in medicine by the end of 2017. And I will stop there.
Thank you very much. Any questions? Tim.
You mentioned that the cancer collection was going to be delayed by a little bit due to the FFPE issue.
Yep. So the pilot's going on right now, essentially to try and... The problem is that inside the NHS, the process, the SOP is around fixation.
Yeah.
Now, if you want to change to fresh frozen, that's a major change inside of health service protocols. It will be expensive. So it doesn't seem to be... It's not necessarily the problem with fixation, it's the problem of extraction of the DNA out of fixation, and whether that can be optimized in a way. And so there's parallel investigations of that going on at the moment.
Have you got an idea of when you'll have an answer to that query? So
Quite
the cancer
soon.
collection, I think, was going quite soon.
Yeah.
Okay.
I mean, now that the GMCs are all signed up, this has to become operationalized very, very quickly. So,
Can you say something about the basis on which you're assessing the second phase of the annotation suppliers? I mean, you mentioned scale already in terms of can they scale up, but one presumes they also have to be assessed in terms of sort of accuracy, validity, and so on and so forth.
Yeah. So, yes, basically, we will run. We have several things running in parallel in the sense we actually need to do some interpretation of the samples we've already collected. I mean, we're going to be collecting at 100 a day by the full capacity of this project, and we have to return those with whatever analysis we can get at that stage. In parallel, we want to assess relative performance. So we're going to run various things in parallel and assess them as we go along.
Okay, 2 more questions, Anne,
Yep.
and then one at the back.
Could you say a little bit more about how individual patients will be selected and enrolled in this? And will this vary somewhat between the different participating partners? And is it going to be mainly large teaching hospitals that are the source of all these patients?
So those are the GMCs, the Genome Medicine Centres. So there's that list, and it does give pretty close to broad geographical coverage. Obviously, though, most of those are not one organisation. It's a consortia of hospitals that have got together and they've, in their specific, it's a very, very detailed specification managed by NHS England, which includes all of them having to essentially say how many patients in the different rare disease areas and cancers they anticipate collecting. So all of those numbers are in their bids, essentially.
So there will be consistency?
Yes, I mean, I think it will vary according to, specialities of different hospitals, but I think it'll be broadly consistent.
And is there a penalty if they don't meet the numbers?
Yes, executed by NHS England.
Okay.
Well, I think, they're being paid per sample, essentially. But, yeah, there's a heavy hitter managing all that.
Okay. The last question there, because we are running
Yep.
out of time.
In the 11 GMCs, will they use the same SOP for the extraction from the FFP samples, or will they choose their own methods that they, for example, use currently?
No, I think that's part of the evaluation. I mean, that's one of the problems with this, that you actually want to have consistency across the country, and we want to establish common SOPs.
And that's what they're doing at the minute, is the
So
evaluation.
at the moment, there's basically the question is, do we have to move to fresh frozen,
Right,
which
okay.
will require, I mean, the GMCs, that's a topic of discussion with the GMCs right now.